think that should work.
Yeah. Great. Thanks, Ben. So I'm very happy to introduce Susan Magnoli today. Susan comes to us from Michigan State, where she did her PhD with Jennifer Lau on the evolutionary ecology of legume-rhizobia interactions. And she wrote and received a USDA postdoctoral fellowship, and then wrote a supplementary grant to the EPSCoR maps, both of which allowed her to look at the effect of land use and climate and on the effects of on the legume-rhizobia interaction. And then she's also been working on the interactive effects of legume-rhizobia and AMF. And that's something that is being worked up into another proposal that Susan is also writing. So Susan, take it away.
Thanks, Jim. Thank you all for coming. Both have some in-person people and on Zoom. I'm excited to be sharing some of my stuff today. So like Jim said, I'm going to be talking about a couple of the things I've been working on as a postdoc here, focusing on the factors of plant microbial mutualist interactions.
Just do a quick check to make sure everybody can hear Susan okay out in Zoom land, because here in Noguchi we don't know because we can hear her. Can somebody just maybe type in the chat? Oh, looks like lots of thumbs up.
Okay,
So,
awesome.
Thanks.
Cool. Okay, so microbial mutualists play a critical role in plant fitness, particularly resource mutualists such as mycorrhizal fungi, nitrogen-fixing bacteria can have huge effects on plant fitness by helping plants acquire resources that are essential for growth, such as phosphorus and nitrogen. And because they can have these huge fitness benefits, they've been shown to actually influence plant population dynamics, plant community composition, and even ecosystem functioning. And so understanding the context dependency of the outcomes of these mutualisms can be really important in ecology. And so today I'm going to be talking about two projects in particular that I've been working on as a postdoc, both focusing on plant microbial mutualist interactions. So first I'm going to be talking about a project focusing on the legume-rhizobium symbiosis, looking at legume responses to rhizobia communities from grasslands during land use history. And then for the second part of the talk, I'll focus more on some recent work we've been doing on the legume-rhizobium-AMF tripartite mutualism. Okay, so first, rhizobia. So rhizobia are bacteria that live in the soil, and when they encounter the roots of a compatible legume species, they trigger the plant to make root nodules to house the rhizobia. And in those nodules, the rhizobia fix nitrogen. That is, they take atmospheric nitrogen and convert it into a form that plants can actually use. They give it to the plant in exchange for carbon from the plant. And under the right environmental circumstances, rhizobia can be hugely beneficial to legumes because they essentially let them access this big pool of nitrogen that's inaccessible to most other plant species. And in turn, the rhizobia benefit from the plant. So we have this kind of nice classic mutualism. But we know that there can also be a lot of variation in the outcomes of this mutualism, depending on the legumes and the rhizobia that are participating. So from a plant perspective, we know that we can see variation, partly because legume species can vary a lot in their dependence on rhizobia. So for example, highly domesticated crop species, such as soybean, have been shown to have lower responses to microbes in general and to rhizobia than their wild ancestors, probably as a result of inadvertent selection against mutualism traits during the domestication process. But this isn't totally limited to domesticated species. We know there's just natural variation between plant species and also between genotypes within a species in how much they benefit from associating with rhizobia. And we also know that legumes can vary in their rhizobia specificity. So rhizobia are a paraphyletic clade of bacteria in about 13 genera. And some legume species will only associate with one genus or even one species of rhizobia, whereas other legume species have been shown to associate with multiple genera. And so not all rhizobia are everywhere in the landscape. So as a result, rhizobia have been shown to influence legume species ranges. So if a legume is expanding its range and encounters a habitat where there are no compatible rhizobia, they might not successfully establish. And so this in turn can influence plant community diversity. And we also know that variation in this mutualism can arise from the side of the rhizobia. And for the purposes of this talk, I'm going to focus on kind of how land use effects or human alteration of the landscape can affect rhizobium quality and community composition. So we know that chronic nitrogen deposition can influence the quality of rhizobia. So in this study, trifolium clover plants were grown with rhizobia harvested from fields that had not been fertilized, and with rhizobia from fields that had been fertilized with nitrogen at agricultural levels for about 20 years. And they found that rhizobia from the fertilized fields were much less beneficial than rhizobia from unfertilized fields, suggesting that the nitrogen addition had caused rhizobia to evolve to be more parasitic and less mutualistic. So we know that our agricultural practices can have a big effect on rhizobium quality. And we can also speculate on how land use and disturbance in particular might affect rhizobium community composition. So when we disturb a site and we remove host plants, probably that's going to affect the rhizobia community. Although rhizobia can live and be free living in the soil, we don't know for sure how long they kind of stick around if their host plants aren't there, or whether they maintain the ability to fix nitrogen over time. So it seems like human activities and alterations can probably have big effects on rhizobia communities. So thinking about all this variation in the mutualism kind of leads me to my project here, looking at whether this mutualism can act as a driver of grassland composition. And so this work was somewhat inspired by some observations that we see here in prairies. First, that disturbed and restored prairies often lack native late successional plant species. So late successional species are the slow growing, really long lived species that we typically find in high quality remnant prairies. And we also observe often that remnant prairies have fewer early successional or weedy and invasive species. And so this observation kind of holds true, not just for legumes, but for many species in general. And we know from previous work in the Bever Schultz Lab that the microbial community has something to do with this, specifically the AMF community. So Liz Kolgel's work and others in the lab have shown that plant responsiveness to arbuscular mycorrhizal fungi depends on plant life history. So the late successional species that are kind of emblematic of remnant prairies are often much more responsive to AMF than weedier species are. And we know that disturbance of these grassland systems alters the AMF community in ways that make it harder for late successional species to establish in disturbed sites. And so for my work here, we really want to know, is the same true for rhizobia? And so it might be reasonable to expect that because rhizobia are also microbial mutualists, we might see the same patterns in responsiveness in these kind of late successional versus early successional or weedy species. And we might see similar reactions to disturbance. Although there are some biological differences between rhizobia and AMF that might make the response to disturbance a little different in rhizobia. So like I mentioned before, rhizobia can be free living in the soil, unlike AMF, which kind of die out when their host plants are gone. And so maybe rhizobia communities might be more resilient or resistant to disturbance. They're also probably less dispersal limited than AMF. They're bacteria, they're very, very small, unlike AMF spores. So they might be a little easier for them to colonize disturbed sites after disturbance. But we just kind of wanted to explore these things with rhizobia. So my two main questions for this project were, first, does legume response to rhizobia vary with plant life history? And then, does legume response to rhizobia vary with land use history? And kind of inherent in that question is, does land use affect rhizobia communities? And so to approach these questions, I did a series of greenhouse growth experiments. And the first few experiments were focused on legume response to the soil microbial community as a whole, so not just rhizobia, but the whole microbial community from sites with different land use histories. And so this work is somewhat tied into the microbiome of aquatic plant soil project, the MAPS project, which many of you are involved with or at least familiar with. And so through the MAPS project, we have soil samples from sites all across Kansas. And from each site, we have samples of the soil from different land use histories. So we have samples from remnant prairies, post-agricultural grasslands, and agricultural fields from all these different sites across Kansas. And so for my experiments, I grew a bunch of different legume species kind of falling into these three life history categories. So we have native late successional species, native early successional species, and then non-native invasive species. And so we grew these plants in the greenhouse in sterilized soil and inoculated them with small amounts of the live soil collected from these sites. So we assessed plant growth and nodulation, that is how many root nodules they made to house rhizobia. And I ended up doing several experiments for this, basically because we wanted to look for generality across sites and also across plant species, but to do that all at once would have been way too many plants in the greenhouse at once. So I ended up doing a series of experiments. So in some, I inoculated a small group of focal legumes with microbial communities from many different sites. And in another experiment, I inoculated many legume species with microbial communities from just one site. And so because I did this experiment several times, I ended up analyzing data using meta-analysis techniques to look for generalities across both kind of sites and species. And so what we found generally from these experiments were that biomass and nodulation vary with plant life history. So this first graph is showing the results of our meta-analysis of biomass. And so we have our three plant groups on the x-axis, and the different colors represent the different microbial communities. And our response variables here are log response ratios, comparing inoculated plants to uninoculated controls. So a positive response indicates that inoculated plants grew better than uninoculated plants, which you can see is the case for all of our plant groups in all of our microbial treatments. But the main thing that popped out in this analysis was that our two native plant groups, so the late successional native and the early successional native, tended to respond more strongly to microbial communities than the invasive group. So basically, there's a bigger difference between inoculated and uninoculated plants in the native groups than the invasive ones. And interestingly, this response didn't seem to depend on where the microbial community came from. So we didn't really see any significant differences in that. But the overall differences in responsiveness between plant groups was there. And then we also looked at the number of root nodules that plants made. And so again, this was a meta-analysis response, and I had to construct an effect size. And we don't really have a traditional control in this analysis because the control plants didn't have any nodules. So I made the plants inoculated with the ag microbial community, the control level in this analysis. And so I'll walk you through exactly what that means in terms of what you're seeing here. So for the late successional plant group, we see positive values for post-ag and remnant microbial communities, which indicates that they made significantly more nodules with those two microbial communities than with the ag microbial community. In contrast, we see that the early native group and the invasive group, these values don't really differ significantly from zero, indicating that they make the same amount of, roughly the same amount of nodules with each microbial community. So this suggests that there is possibly something different between microbial communities from these sites, at least in ways that the late successional plant group seems to care about. Although that doesn't really translate into a growth effect that we're seeing in the biomass results. But again, this is plant response to microbial community as a whole, not just rhizobia. Okay, so Naturally, our next step was to just look at plants' response to rhizobia. So in all of those prior experiments, when I harvested plants, I was also harvesting root nodules that I took back to the lab and cultured to isolate rhizobia strains. So I built up kind of this big library of strains harvested from each plant species, from each site, from each land use type. So I have a lot of rhizobia strains. But for this experiment, I decided to use strains from just one site with the 3 different land use histories. And I took 2 strains isolated from each plant species from each land use type. And then I would mix together all the strains from remnant, all the strains from post-ag, and all the strains from ag to make these 3 different treatment mixtures. And we kept the cell density constant for each strain. So we had the same amount of each strain going into each mixture. And then I inoculated the same big group of legumes with those strains in the greenhouse. And we also provided all plants, even the controls in this experiment, with AMF, a mixture of native AMF, just because these plants are almost always growing with AMF in nature, and they can have big effects on plant growth. So really the only thing that differed was the rhizobia treatment. Okay, and so we found here that biomass and nodulation again varied with plant life history, and also to some extent with rhizobia. So these results are just from one experiment, so I didn't have to do a meta-analysis. So here I'm just presenting treatment means. And so the thing that we see again here is that our 2 native groups respond more strongly to rhizobia than the invasive group. That is, there's a bigger difference between the inoculated plants and the controls for the natives, but not for the invasive group. And again, we see that with the natives, they don't seem to care too much who they're inoculated with. The rhizobia origin didn't have a significant effect on plant biomass for these 2 groups, but it did for the invasive group. So we see that rhizobia inoculated with, plants inoculated with rhizobia from ag sites in the invasive group significantly outperformed control plants, with the other 2 groups kind of in the middle. So generally, invasives responded a little bit more to rhizobia from the 2 disturbed land use types than the remnant. Okay, and in terms of nodulation, again, with the 2 native plant groups, we don't have any significant differences between rhizobia types. So they made the same number of nodules regardless, generally, of who they were inoculated with. And the invasive group produced significantly more nodules with the 2 disturbed site rhizobia than with the remnant rhizobia, which kind of mirrors to some extent the biomass results we're seeing for the invasive group. Okay, so thinking back to my original questions for this project, does legume response to rhizobia vary with plant life history? We see generally yes. So both response to rhizobia, which is what I just showed you, and response to the microbial community in general are stronger in our 2 native plant groups than in the non-native invasive group. And I thought these results were interesting for several reasons. First, kind of one of the many hypotheses for why invasive species might be successful invaders is that they might be less responsive to mutualists. So they go to invade a new site, and maybe compatible mutualists aren't there, but they don't really care because they don't really need those mutualists. And so I think these results kind of support that to some extent. But we were somewhat surprised to see that successional stage, so either late or early successional natives didn't differ between each other. And that was surprising based on what we know about mycorrhizal responsiveness in these groups. So this graph shows, it compiles a bunch of data from about 200 plant species, not just legumes, but legumes were included here, looking at mycorrhizal responsiveness by successional stage. And so generally in the late native successional groups, we see high mycorrhizal responsiveness, but early successional and non-native groups tend to have about the same, similar responses to mycorrhizae, which is not what we're seeing with the rhizobia. We're seeing kind of equal responses, and it's kept more native versus invasive and not really by successional stage. And so I think this is interesting, and it could be kind of fun to think more about why we might be seeing these differences. Okay, and then does legume response to rhizobia vary with land use history? We found to some extent, it seems like mostly we see variation in the invasive group with them responding more strongly to rhizobia from disturbed sites, which is interesting. And we were again a little bit surprised by these results, given what we know about mycorrhizal responsiveness in the late native species group. So we know that this group of species tends to respond very strongly to mycorrhizae from remnant prairies, which is not really what we're seeing here. And so maybe there's just some inherent difference between rhizobia and AMF. But we're also thinking about whether kind of the timing of these experiments might have an effect on this. So these were all greenhouse experiments, so are fairly short, matter of months. And late successional native species are very slow growing. And so we're wondering if maybe we're just not seeing effects because these experiments were so short. And do have some evidence that this might be going on from some field work that we've been doing. So we also wanted to look at legume-rhizobia interactions in a more natural setting in the field. And so in spring of 2020, we set up an experiment with 3 focal legumes, so Amorphocanescens, late successional species, native species, Chamichrista fasciculata, an early successional species, and Lespedeza cuneata, an invasive species. And we grew these seedlings in the greenhouse and then inoculated them with either postag rhizobia or remnant rhizobia, and then planted them out into kind of a mini restoration out at the Land Institute. And so at the end of the first growing season of this experiment, we saw no effect of rhizobia on survival or growth of any of these species. And Chamichrista is an annual, so it was done after the end of the first growing season. The Lespedeza we removed from the field because I didn't want to be responsible for starting an invasion on that part of the Land Institute property. But I did leave the Amorpha in the field, and I've been tracking them ever since. So initially, we didn't see an effect of rhizobia on Amorpha survival, but then I surveyed them this year, and we're starting to actually see something interesting. So we found for year 2, Amorpha that had been inoculated with rhizobia from remnant prairies had significantly higher survival than uninoculated controls. Plants that had postag rhizobia did a little bit better, but they weren't significantly better than controls. So I think this suggests that there could be kind of a time issue that we're running into in our greenhouse experiments. And I think it would be cool to continue some of this work, maybe in more natural settings like this one, or for longer time scales to really kind of see what's going on with kind of these native versus disturbed rhizobia. Okay, so that is my rhizobia, highly rhizobia focused part of the talk. And now I want to move on to our work that we've been recently looking at context dependency and synergistic outcomes of these tripartite mutualisms. Okay, so far I've been mostly focusing on one pairwise mutualism between legumes and rhizobia. But we know that in nature, plants interact with multiple mutualists simultaneously. So a legume that's associating with rhizobia is probably also associating with pollinators. They could be associating with ants in a defensive mutualism. They're probably also associating with AMF. And so all of these partner mutualists have the potential to interact with each other either directly or indirectly in ways that affect the ultimate outcome for the plant host and make the outcome for the plant host somewhat harder to predict than a pairwise mutualism. And so one potential outcome of a multi-mutualist system is synergistic benefits for the host, which essentially benefits when you associate with multiple mutualists, benefits are greater than you'd expect based on additive expectations of the effects of mutualists alone. So I think it's kind of a complicated definition, and I think it's easier to think of it graphically. So say we have a system with a plant and 2 symbionts. When a plant is growing alone with no symbionts, it'll have however much biomass. When it's grown with symbiont 1, biomass increases by a certain amount. Same with when it's grown with symbiont 2 alone. And if the effects of both symbionts simultaneously are additive, we would expect plant biomass to be at this dotted line here. That's the additive. It's a combination of those two effects. Any plant biomass above that additive expectation is what we define as a synergistic benefit. So basically non-additive benefits from these mutualists. So we have a nice real-world example of this from Anna Larimer's work on amorphokinesins associating with AMF and rhizobia. So when plants associate with AMF alone, they do a little bit better. It's hard to tell with the scale of this graph, but they do a little bit better than when associating with no symbionts. Same with rhizobia, they do a little bit better. But when they're associating with both symbionts simultaneously, they get this huge growth benefit, which is synergistic. And so why does synergism happen in this system in particular? We hypothesize, at least, that synergism is going to arise in systems like this when symbionts provide complementary limiting nutrients to the plant. So a legume associating with rhizobia is going to get nitrogen from the rhizobia. We know that AMF provide many different nutrients to plants, but primarily phosphorus. So a legume associating with AMF is going to get increased phosphorus, and nitrogen and phosphorus are, of course, both essential nutrients that are often limiting for plant growth. And so when legumes are associating with both mutualists, we expect them to neither be particularly N or P limited because they're getting those resources from the mutualists. And so this allows plants to increase their growth because they have both of their essential nutrients. But we predict that it also lets plants kind of reinvest in mutualists. So nitrogen fixation by rhizobia is a pretty phosphorus intensive process, and AMF do require some nitrogen. So not only can plants use these increased resources to increase their growth, but they can reinvest in their mutualists, thus generating more benefits and increasing their growth more, generating synergism. Okay, so we know that most legumes associate with both rhizobia and AMF. So naturally, we kind of want to know generally, do these interactions lead to synergistic benefits? And so Anna Larimer again did a meta-analysis looking at this back in 2010. And interestingly, the answers seem to be no. We don't generally see synergistic benefits in this mutualism. In fact, the outcomes tend to be either additive or slightly less than additive. And so naturally, we kind of want to know why. And one thing that pops out of this particular data set that they used was that most of the data came from studies of annual plants and a lot of crop species, which we know generally tend to be less responsive to microbial mutualists than longer lived species. And so thought that maybe that could be an explanation for why generally we're not seeing synergism in this mutualism. So we recently did another meta-analysis. So this was started by Silmar Premieri, who studies legume-AMF rhizobia interactions in Brazil, and he was up here as part of a sabbatical visiting Jim's lab. And so we rescanned the literature. So this was 10 years later, and we were able to find a bunch more studies of both annuals and a bunch more perennials to put together kind of a bigger data set. And a lot of these papers came from the Portuguese language literature too, which was great. And so overall in our meta-analysis, we found that as predicted, perennials were more responsive to mutualists in general and experienced synergistic benefits while annuals did not. So this graph is showing our main kind of biomass results. And so let's focus on these first two bars here. This is a log response ratio comparing biomass of plants grown with AMF to plants without AMF. So the positive values indicate that AMF increases plant growth and to a larger extent in perennials shown in gray than annuals shown here in white. We see the same response for rhizobia, comparing plants with rhizobia with plants without rhizobia. And then these synergism bars here are where, so a positive synergism value indicates a greater than additive effect of associating with both mutualists, which we see is true for perennials, but not for annuals. And we also wanted to use this data to see if we could find evidence for our hypothesis that resource complementarity can drive these synergistic outcomes. And so we approached this by looking at the nutrient concentrations, particularly N and P tissue concentrations of the plants in some of these studies. And so you can imagine that plants associating with only AMF are going to have decreased nitrogen to phosphorus ratios because they're getting phosphorus from the AMF. We would imagine the opposite to be true with plants associating with only rhizobia. So we would expect nitrogen to phosphorus ratios to go up because they're getting additional nitrogen. And for plants associating with both symbionts, we would expect kind of an intermediate nitrogen to phosphorus ratio, indicating that they're not accumulating or being limited by either of these nutrients. So a subset of the data in our meta-analysis did provide nutrient content. So we looked at that, and we generally found our predictions to be true. So plants associating with only AMF had decreased nitrogen to phosphorus ratios compared to plants without AMF. Opposite for rhizobia, and then we saw these intermediate values for plants associating with both symbionts. So we took this to be a sign that our predictions about resource complementarity could be true, or that resource complementarity could potentially be driving these synergistic outcomes. And we also wanted to look at whether increased investment in symbionts could also be helping to drive these synergistic outcomes. And so this graph is, the x-axis is quite different from the previous ones I've been showing you. So I'll walk you through it here. But basically, we looked at investment in symbionts in terms of nodule number, nodule biomass, and AMF colonization. And so we found for plants that associated with both AMF and rhizobia, they had higher nodule numbers than plants associating with only rhizobia. So basically, when plants had access to AMF, they also made more nodules. We see the same general pattern for nodule biomass, but it wasn't statistically significant. In contrast, when we look at plants associating with both symbionts compared to plants only associating with AMF, we don't see an increase in AMF colonization. So basically, associating with rhizobia doesn't make plants further invest in AMF in terms of colonization, at least. And so this kind of partially supports, at least looks like when you have AMF, you can invest more in rhizobia, which supports our hypothesis, but the opposite doesn't seem to be true, at least with this data. But we can also look at this in terms of the correlations between investment in symbionts and synergistic outcomes. So these graphs show basically the bars that I was just showing you in the previous graph. So we have nodule number, nodule biomass, and AMF colonization correlated with the biomass of dual inoculated plants. And we generally see statistically significant positive correlations between investment in symbionts and synergistic outcomes. So that does suggest that increased investments in symbionts could partially be driving synergistic outcomes. So in general, we were really excited about these results. That was pretty cool. But one caveat with this data set is that we still, so one strength of this data set we thought was that we do have a lot of data from perennial species, which was lacking from the previous meta-analysis. And we have a lot of data from many different annual species, but almost all of these annual species are domesticated crop species. And they are many of the data points in our data set. And so this isn't super surprising because a lot of the literature we were pulling these from are studies trying to figure out how we can use microbial symbionts to increase crop production instead of dumping on more fertilizer. So that's cool. Glad all those studies are out there. But it does kind of make it hard to separate the effects of being annual from being domesticated in this study. We do know from studies of many prairie species that annual or shorter-lived plants do tend to be less responsive to microbes than perennial plants. And so we don't think there's totally crazy to say that there's differences between perennials and annuals, but there's clearly a little bit more work that needs to be done here to kind of tease apart the effects of plant life history here. And so thinking of that, I am working on a proposal with Jim and Tom Platt at K-State to kind of dig more into the context dependency in these interactions. And so what we're planning to do is to explore additional factors that might influence synergism. So particularly abiotic conditions, looking at soil nitrogen and phosphorus and how variation in those levels can potentially influence synergistic outcomes. We want to delve more deeply, like I mentioned, into plant characteristics. So not just looking at perennial versus annual, but looking at successional status, native versus non-native, domesticated versus undomesticated. And then we'd also like to look a little bit into microbe quality, which would tie in nicely with a lot of the work in the Bever Lab on land use effects on microbe quality. And then we'd also like to further explore the mechanisms potentially underlying synergism. So if we're able to do all these experiments, manipulating all these factors, we can also measure plant nutrient concentration to get a more robust data set to look at this kind of testing this nutrient or resource complementarity hypothesis that we have. And then finally, we'd really like to develop theoretical models and predictions for when we might expect synergism, kind of with the big picture goal of thinking about like how we can maybe potentially manipulate plant microbiomes to, better fulfill our management or production goals in these systems. So with that, I'd like to thank all my collaborators and my funding sources, and I would be happy to take any questions.
Yay. Wow,